What you see on the screen here, we've looked at ways in which animals can protect themselves against predation. Here are two examples. You have a caterpillar here of a moth. It's masquerading as a twig. So it's a twig mimicking or twig masquerading caterpillar. Here you have flowers of an oak species. This one here is a caterpillar. This caterpillar is masquerading as oak flowers. If you look closely, you can see that the caterpillar has been feeding on these leaves. What is even more interesting is that both these caterpillars belong to the same species. Belong to the same species, Nemoria arizonaria in North America. And what is even more surprising is that you can take a bunch of eggs from the same female, you give them only oak catkins. Catkins are a type of inflorescence. So if you give them oak catkins, they feed on them and develop into this kind of caterpillar. On the other hand, if you feed them leaves, they grow into this kind of morph. You can even produce clones. Produce a bunch of clones, grow half of them on oak catkins, they'll turn out to be this. Other clones, if you grow them on this, they'll turn out to be this. And this variation in phenotype and its dependence on diet has also been shown in artificial diets. So what researchers have figured out is that it's related to how much tannin there is in the diet. So when they've grown these caterpillars on artificial diets with high tannins, they develop into this kind of morph. In nature, this butterfly, remember it's found in North America, it's a temperate butterfly. It overwinters, in other words, it hibernates, and it has one generation when the spring comes up. This is a spring morph. So during this generation, oaks have catkins. So their host plant is oak trees. So in the first generation of the year, they feed on catkins, develop into this morph, and you expect that they are cryptic, so they benefit from masquerade. These guys go on to develop into adults and mate, lay eggs. By the time the next generation of eggs are growing up, once they hatch and the larvae come out and they're growing, they feed on leaves. So once they feed on leaves, they develop into this morph, and this presumably gives them a survival advantage because they are masquerading as twigs. So remember that in this condition, when this morph is found, there are no catkins. So looking like catkins may not help as much as it would have helped in the spring generation. So what researchers have found is that it's the diet, it's specifically the tannin content which decides whether they develop into this morph or this morph. So even in the spring generation, If you were to give them only leaves, they would develop into the summer morph, this kind of morph. Similarly, during summer, if you are somehow able to give them catkins, they would develop into this, they would develop into a catkin morph. So all these experiments have been done. What do you see here? You see that the same genotype can develop into either this morph or this morph. In other words, the same genotype can express two very different phenotypes, and this is dependent on the environment. The environment in this case is the diet. What we have seen is an example of phenotypic plasticity. Phenotypic plasticity is the ability of the same genotype to express multiple phenotypes depending on the environment. Phenotypic plasticity can involve any phenotype. It can be morphology, physiology, behavior, and so on. And it can be a response to any abiotic or biotic environmental parameter. Here's one more example involving an insect. Arachna levana is found in the Palearctic region in Europe and Northern Asia. This butterfly has two morphs. So the adults in the spring generation look like this. They have bright orange and black, whereas in the summer generation they look like this. Again, as in the case of Nemoria arizonaria, this is a generation that comes out of hibernation first, and the eggs laid from this generation, they develop into this morph. In this particular case, experiments have shown that the environment involved is photoperiod. In the spring generation, day length is shorter compared to the summer generation. So remember that the longest day length is sometime in July. That's when midsummer is celebrated. It's approximately at that time that midsummer is celebrated. Yeah. So during spring, the developing larvae experience short day lengths and they develop into this morph, and during the summer they experience longer day lengths and they develop into this morph, and this can be replicated in the lab. If these are grown in short day length condition, you get this morph, and if you grow them under long day length conditions, you get this morph. In our own backyard, we have a famous example of phenotypic plasticity in wing patterns. You have this plasticity in the presence of eye spots. So here is Bicyclus aninana. This is an African butterfly, but the phenomenon which I am going to talk about is there in many tropical butterflies, including a lot of butterflies found in India. So a lot of these butterflies in the wet season, they have these conspicuous eye spots which are found along the wing margins and are serially arranged. So we have discussed how these eye spots can protect the butterfly against predation by deflecting attacks away from the body towards the wing margins. In the dry season, they have this morph. So this morph is cryptic. It's cryptic through background matching. In the dry season, you have a lot of dry leaf litter on the forest floor or whatever else is their habitat. So there's a lot of dry leaf litter. So the butterfly is likely to be viewed against a background consisting of predominantly brown. Therefore, having no eye spots and having predominantly brown, basically it has different shades of brown, the butterfly can benefit through background matching and perhaps also disruptive coloration. On the other hand, in the wet season, the background is predominantly green. I must mention that these butterflies are predominantly found close to the ground. So during the wet season, this butterfly is likely to be found close to green vegetation, grasses and so on, and therefore it's likely to be viewed against a green background. In that case, it will not benefit from background matching, and it benefits by having eye spots, deflective eye spots. This has been shown in experiments. These butterflies, the dry season morph and the wet season morph have been exposed to predators under lab conditions, and these experiments have shown that under the dry season conditions, basically against a leaf litter background, dry leaf litter background, this morph performs better. And other experiments have also shown that having eye spots is better when the butterfly is not background matched. In this particular butterfly, Bicyclus aninana, the environmental cue or signal that makes them develop into either this morph or this morph is temperature. So this is an African butterfly, and in the regions where it's found, well, not all regions, but in Malawi, so these experiments were done on a population collected from Malawi. So the experiments I'm going to describe. And in Malawi, during the dry season, it's cooler, it's colder. During the wet season, it's warmer. And from a lab population established from founders from Malawi, so on this lab population, experiments have shown that if they are grown at high temperatures, they develop into the wet season morph. If they are grown at low temperatures, they develop into this morph. So in nature, when they develop under cold temperatures, it's probably an indication that the adults are going to face a dry season environment, and therefore they develop into this morph. Similarly, when they grow under high temperature, when the caterpillars are developing under high temperature, it's an indication that the adults are going to face a wet season environment, and they develop into this. Remember that all of this has evolved. The butterfly, the larvae are not making a decision by thinking. It just happens. There are the genes, and there are some genes that are sensitive to the environment, and they take on either this pathway or that pathway. So you have two developmental pathways, and either one of the pathways is triggered as a response to larval growth temperature. Interestingly, in this species, Bicyclus aninana, the plasticity is not restricted to wing patterns. There is also plasticity in their behavior. In the wet season, females exert selection pressure on male traits. So experiments have shown that in the wet season, females choose males based on this central white spot. So this central white spot is UV reflective, and experiments have shown that there is stabilizing selection on this spot. So females don't like butterflies which have really large spots, neither do they like males which have small spots. In the dry season, the roles are reversed. The males become choosy, the females are not choosy. Again, the males are using the central UV reflective spot as a signal in sexual selection. So it's a very interesting experiment. The same environmental cue, temperature, can give you either this morph or this morph, and when you look at the behavior of the two morphs, you find differences in sexual preferences. It's not really clear why they have these differences across seasons, but yet it's a very fascinating phenomenon which they have found. In this barnacle, there are two morphs. This is one morph where the aperture or the opening is perpendicular to their substrate. But when the predator is present in the water, so one of their predators is this sea snail. When the predator is present in the water, they detect the presence of the predator and they develop into this morph. In fact, even if you remove the predator, but if the predator has been there for a while, the predator leaves some chemical signals which can be sensed by this barnacle during the developmental stages. So again, these chemicals left behind by these predators are a signal. Some genes are responding to the signal and they trigger either this developmental pathway or the other one. And well, I forgot to mention that this morph, where the aperture is perpendicular to the substrate, is more resistant to attacks by the predator. So that is an adaptive advantage of having this morph. Now, you can ask this question: if this morph is better protected against predation, why does this morph exist at all? Why not always produce this morph? Well, if there is protection against predation, well and good. If there are no predators in the waters, so what? What's the problem? Think about it. Why do you think the snail still has the ability to produce the less protected morph? What might be the evolutionary advantage of having that morph, rather of having the ability to produce that morph? In this graph here, we have environment on the x-axis and the phenotype on the y-axis. For example, this could be photoperiod or this could be temperature. So let's think about Bicyclus aninana. So in Bicyclus aninana, experiments have showed that as temperature increases, the size of the eye spot increases, which is why when they're grown in cooler temperatures, eye spot size is close to 0 and therefore they have a dry season morph. And similarly, when they're grown in high temperatures, eye spot size is very high. So basically what I described was this kind of curve, this kind of relationship between phenotype and environment, an increasing relationship. So this, if you could depict this in a graph, it would be called a reaction norm. Reaction norm is nothing but the range of phenotypes produced by a genotype when exposed to different values of one environmental variable. So you're always talking about one phenotype and one environment. So this is one kind of relationship you can have between the phenotype and the environmental values. You can have a decreasing relationship, or you can also have no change in phenotype at all. So let's look at the number of eyes or the number of wings in a butterfly. If you plot that on the x-axis, and if you plot, for example, temperature, you'll find that the number will remain constant. Therefore, the phenotype coded in terms of number is constant. In other words, in this particular representation, A and B are phenotypically plastic. So phenotypes A and B exhibit phenotypic plasticity. Phenotype C does not exhibit phenotypic plasticity. Polyphenism is a special kind of phenotypic plasticity where you have discrete phenotypes. So in fact, we have already been discussing examples of discrete phenotypes. If you look at the morphs, the spring and summer morphs in Nemoria arizonaria caterpillars or Arachnia levana adults. So these are examples of polyphenism because you have discrete phenotypes. So discrete as opposed to continuous. I'll give you an example of continuous phenotypes in the next slide. Now, another famous example is temperature dependent sex determination in many organisms. It's well known in reptiles. So there's a broad, you already know about chromosome sex determination, chromosomal sex determination, where the chromosome composition determines sex, but in many animals, environmental conditions decide the sex of the offspring. So one of the most widely known environmental parameters that can decide sex is temperature. So basically, temperature dependent sex determination is one type of environment dependent sex determination. So this is known in a lot of reptiles. For example, turtles. They can control the ratio of males to females. A sea turtle, it's born on the beach. A female sea turtle lays eggs, it digs into the sand on a beach, and once the eggs hatch, the turtles come out and they go to the ocean. They remain there for a long time. They feed, they mate, and once the females are ready to lay eggs, they come back to a beach. Now, the female can either dig deep or it can dig a very shallow pit and lay eggs. Depending on how deep it has dug its pit, the temperature will vary. And depending on the temperature, they can be either only males or only females, or at an intermediate depth, you can have an equal proportion of males and females. This is an extreme example of phenotypic plasticity, I would say. And in fact, if you think in terms of the ratio, if you code phenotype as a ratio, then this is not really a polyphenism, the ratio itself is continuous because it can vary from 0 to 1. If you think of female is to male ratio, it can vary from 0 is to 1. But if you code the phenotype in terms of sex, whether it's male or female, then it becomes a polyphenism. Right? So by having this phenotypic plasticity, reptiles can control the sex ratio under conditions where having more males is beneficial. They can get more males, and when females have a fitness advantage, they can produce more females. Therefore, the female that's laying eggs can enhance its own fitness. We have caste in honeybees. In this case, the offspring the larvae that are fed on royal jelly, they go on to become queens, right? And the rest of them become workers. Here, if you code in terms of caste, then that particular phenotype is an example of polyphenism. So how do you get polyphenisms in nature? So there are two ways in which this can happen. One is that the environment itself is discontinuous. So let's assume this is a reaction now of a phenotype for a given environmental parameter. But in nature, the developing animals or plants, they don't experience all of these possible environmental conditions. Let's assume they experience only discrete environments. So either environment ranging from here to here or from here to here. So those that develop under this environmental conditions, they get one phenotype. Those that develop under this set, they develop into this phenotype. When you look at the phenotypes produced, they are discrete. It's not continuous. This intermediate phenotypic values are not observed. Therefore, you get a polyphenism. On the other hand, you can have this kind of condition where the reaction norm itself is discontinuous. So you have a threshold. Beyond this threshold, you get a different phenotype. Below this threshold, you get a different phenotype. So there's a developmental switch. Under this condition, even if the organism experiences the entire range of environments or the entire range of environmental values, you still end up getting a polyphenism. So let's look at this example. I have mentioned the summer and spring morphs of Arachnea lavana. This is an example of polyphenism observed in nature. I also mentioned that the wing patterns react to photoperiod. So under lab conditions, if you were to grow them under a range of photoperiod conditions, so here you have long day length, here you have short day length, and if you varied this in many steps, you in fact get many intermediate phenotypes. But you don't see this in nature because under natural conditions, they either experience this end of the spectrum or this end of the spectrum. So this would be an example for this kind of scenario. In fact, the scenario seems to explain most of the polyphenisms seen in nature. In a lot of cases, once you take them to the lab and grow them under continuously varying environments, you end up seeing the intermediate morphs, and these intermediate morphs may not be seen in nature. But there are known examples of plasticity due to developmental switches. So where you have polyphenism due to developmental switches. It's known in some cases, for example, that there is a hormone signal involved, and beyond a threshold level of hormones you get a different phenotype. And the hormone signaling itself is sensitive to some environmental condition. So in the next slide there's an exercise. I want you to try and find such examples in literature. And of course polyphenisms can develop because of both developmental switches and because of the first scenario that we talked about. It's been shown in Bicyclus aninana that the temperature dependent polyphenism is in fact combination of both a developmental switch and having discrete environments in the wild. So here's the exercise for you.